episode of Oceans Alive for this spring lecture series. And this evening we have, I think, a very entertaining and enjoyable format to present, Science Blunders and Bloopers. And it sounds like a comical title. It is a comical title, but I think many scientific discoveries have not been made because of any deliberate observation. Many are made that way, but many have come out of accidental discoveries. And what we've asked people this evening is to think about some of their discoveries that have come about when they weren't heading down a particular path, but came upon something by accident. And tonight's host will be Dr. Rocky Geyer from the Applied Ocean Physics and Engineering Department here at Hui. And we chose Rocky as a host because he said he's never made a mistake. He's never made a blunder. But he does have the distinct talent to be able to make a story about any data set, whether it's right side up or upside down. And so we thought that was a perfectly appropriate MC for this evening. So Rocky, I'll hand the podium over to you.
I don't think I need the podium. I've got a little microphone. Well, thanks a lot. I've been to one of these. I've given a presentation once before in talking about Massachusetts Bay. And one of my biggest blunders was probably stepping into that hornet's nest of the mix of science and policy in Massachusetts Bay. And this was probably one of the few appreciative audiences that ever listened to me talk about Massachusetts Bay. I'm not quite sure why Judy selected me to do this, but I think it's probably because I have less tact than almost anyone she could think of. So, in case the presenters get dull, I'll probably start pulling in quips from the audience. Well, our first presenter isn't even here, but he sent a video. This is John Farrington, our esteemed Dean and Associate Director for Education. Most of you probably know him. He's been around for a long time. But he was too good at geochemistry, so the administration made sure that he would get bumped up to a position where he couldn't do any more science. He sent me a little info about himself, and his nickname that he gave is Nimble Fingers. And I'm not quite sure why. Perhaps the video will shed more light on this. I doubt it, actually. He says he's a championship typist. I've also heard rumors that this is a very long video. I know that at Jake Pearson's retirement party in San Francisco last year, John made a brief, what was supposed to be 3-minute presentation that lasted about 20 minutes. So don't expect this to be extremely short, and I don't have any idea what John's going to be talking about, but we're going to roll the video and find out.
I'm joining you in person, but I'd like to thank Sherry DeRosa, Tracy Crago, and Judy McDowell for making it possible through Sea Grant for me to be here with you by way of video. And I'd like to thank Tim Silver for being the person behind the camera. When viewing this video, keep in mind the actor and his props that Tim had to work with. This is a story about research and a blooper. At least I like to think of it as a blooper and not a blunder. I leave it up to you to decide in the end. Back in 1972 through 1976, there were several of us here at Woods Hole Oceanographic Institution involved in understanding, or trying to understand, conducting research to understand, the early diagenesis of organic matter in surface sediments, and some others were looking at using organic compounds as traces of processes in the water column. In my particular case, I was interested in lipids in surface sediments, and our colleague Bob Gagosian was interested in using compounds such as sterols to trace processes in the water column. I came from a background in biochemistry and chemical oceanography, and Bob came from a background in organic chemistry and reaction mechanisms. We were both assistant scientists at Woods Hole Oceanographic Institution. You might ask, why were we looking at lipids? Well, first of all, as indicated here in this chart, they've been extensively studied in ancient sediments. They were important in understanding the origin of oil, and petroleum, oil, and gas. And in fact, many analyses had taken place with respect to these compounds in ancient sediments and in petroleum, and for hydrocarbons in particular. Third reason, we had methods for their analyses and detailed structural elucidation. Fourth, we knew they were important biochemicals for various organisms. And fifth, they had a range of chemical and biochemical reactivity which made them of interest in terms of trying to understand various processes in the water column and surface sediments. Just a quick rundown in a nutshell about how we do the analysis. We take a sample such as surface sediment, extract it with an organic solvent, take the organic solvent and separate various lipid class compounds by way of column chromatography. This is basically a column of things like silica gel, and you take various fraction cuts from there. You then concentrate those fractions and analyze them by gas chromatography, which basically separates the chemicals according to their molecular weight or boiling point, with some aspects of their chemical structure also helping in the separation of various compounds. Despite the fact that using gas chromatography, you can often tell by virtue of the retention time, that is how long it takes a compound to go through the gas chromatographic column, as to what its structure is. Many times, especially in those days, and even today, dealing with samples from the marine environment, you have peaks for which you have no idea what the structure is, or those which you would like to confirm the structure. And for that, we use gas chromatography and mass spectrometry. And the mass spectrometry aspect basically bombards the compounds coming from the gas chromatographic column with high-energy electrons and fragments them into characteristic fragments, which by looking at these fragments, you can reconstruct what the chemical structure would be. Or at least that's how it operates in principle and in theory. Some representative compounds at the time of 1972 and 73 that we were looking at were N-alkanes, such as this compound here, carbon atoms indicated by the points in the various stick diagram. This one happens to be an N-alkane, which comes primarily from land plants. In the surface sediments in both Narragansett Bay and Buzzards Bay, and on the shelf off the coast here in New England, we were puzzled by the occurrence of some compounds that had 25 carbon atoms, and we thought might be cycloalkenes. They were very prominent compounds. They were present in high concentrations relative to other hydrocarbons. We couldn't figure out where they were coming from. We were doing analysis of the various fatty acids, and as I indicated previously, Bob and his group were looking at the various sterols, of which cholesterol is one example. One of the problems we had is we could not often, or we had a problem, I should say, in trying to figure out sometimes whether some of these compounds were coming from marine sources or whether they were coming from land plant sources. And trying to figure that out here along the coast was difficult and problematic because we knew we had some inputs from land plants. And so one thought that occurred to us was to go to an area where we had primarily marine organic matter inputs to the sediments, and where the concentrations of the organic matter would be quite elevated, and that would minimize some of our problems in terms of interferences from potential land plant sources, as well as having enough material that we could do analyses and get some structural elucidations. Fortunately, at the time, we came to this conclusion as a suggestion for further research. The Atlantis II was scheduled to be on an extensive cruise in the Indian Ocean. And to get from here to Woods Hole, it was going to go to Recife in Brazil doing some research, across from Recife to Cape Town for Jeff Thompson doing some work on the ridge crest and dredging rocks. And then there was some time that we could have the ship from Cape Town to go up to an area off of Walvis Bay, Namibia, the old Trust Territory of South Africa, and sample in very organic rich surface sediments. There's a very strong upwelling area here, a desert inshore, so relatively little input from land plant materials or runoff, organic carbon concentrations up to 20% in the surface sediments. So off we went on A2 cruise 93, leg 3. One of the notable aspects of this cruise, which is an entirely different story to be told at some point in the future, is the fact that we were at sea over Christmas and New Year's between December 1975 and 1976. And furthermore, as we went up and took some samples up here and did some geophysical transects up here for some of our folks on board, we ran into a Cuban and Russian fishing vessel operation, which was rather mysterious and going on during the Angolan War. Another story. So as we extracted the surface sediments after we got back to Woods Hole and began the analyses, we ended up with a number of hydrocarbon fractions from the analytical chemistry runs that we wanted to further look at with respect to their structures. And so we submitted these samples to the GC Mass Spec facility, which had been set up and was running with the great input and expertise of Nelson Frew. This was in the first floor of the Redfield building, and Bob's laboratory and my laboratory were on the third floor of Redfield building. Enter the story a summer student employee by the name of Dan Rapetta, who came to my laboratory as an undergraduate between his junior and senior year. And one day, Dan was interested in figuring out how we use GC Mass Spec for structural elucidations. So I took him into the lab, downstairs into Nelson's lab, and we sat in front of the computer where the various files had been stored from the GC Mass Spec system. And I began by telling Dan how we could go through and search the files and find signature fragment ions for various types of organic compounds. And I started off by saying one of the most common compounds that's analyzed in oil, for example, would be steranes. And we could use a fragment ion by 217 atomic mass units and look at its relative abundance in the sample and begin to focus in on seeing how many steranes there might be there and then do structural elucidation. Then I said, but of course, in surface sediments, unless it was polluted with oil, and that's not the case as far as I know off of Walvis Bay, then we would not be able to find the steranes. And if we put in the 217 fragment ion and asked the computer to search for its relative abundance, we'd find a lot of garbage, basically, and no signal. And I said, but let's try it out and see what happens. And much to my chagrin and excitement later on, as we plotted this out, and this is the ion current at the top, we have a signal going along here, which is the detector signal, and then Nelson changed the sensitivity range here, and we have this is a total ion current. It's very difficult to see that there are any compounds there present. But then we looked at the 217 plot, whammo, 3 peaks were sticking out there. Now, I had looked at these samples before, so I was pretty confident, because when I looked at them before, I had seen this particular signal down here, which you see is 217. You don't have very much in terms of the 3 peaks sticking out. Comparing the two together, you can see the difference. So I said to myself, what the heck is going on here? Well, the first thing that's going on is the following. After checking, I found out that the sample I looked at before was JWF6. That's the file. In my nimble-fingered hunt and peck method of typing, had inadvertently typed in JWF7, which was a sample that we had subjected to hydrogenation, to add hydrogen across the double bonds, to look for those mysterious C25 compounds that we thought were present in the Walvis Bay sediments, that were the unknown compounds we were trying to figure out in the Buzzards Bay sediments. So to make a long story short, what we had seen here was the presence of steranes, which shouldn't be present in the surface sediments, and could only be there as a result of hydrogenation if there were sterines, unsaturated hydrocarbon sterile diagenesis products. To our knowledge, this had only been found in ancient sediments and on its way to becoming oil. And so we were quite excited about this. We printed out a couple of the mass spectra of the sterines, went running up to the third floor of Redfield and stopped by to see Cindy Lee, who was a postdoc with Bob Gagosian and I at the time, working on sterile geochemistry in the oceans. And we talked it over with her, and she said, sure enough, I know that particular mass spectra right there. That's a cholestodiene. And she pulled the paper out, and we looked at it, and sure enough, there it was, a cluster dying. With that in hand, I took the data, preliminary data as it was, across to see Bob Gagosian, and I said, Bob, take a look at this. And as we went over the data, Bob became very excited. Now, those of you who know him as director knows that he gets very excited about a lot of things, but believe me, this was a greatly excited Bob Gagosian. Unfortunately, I cannot share with you all of the words that he said. Tim would probably have to edit the tape. But Bob was really excited. I was excited. We really got turned on. We cranked through many more sediment samples. Bob and Nelson worked through the GC mass spec stuff. And we put together my knowledge from my group's interactions and understanding the diagenesis of organic matter in surface sediments, and Bob's understanding of the reactivity and mechanisms of organic chemistry, and came up with a diagram of diagenesis of sterols, indicated by this particular compound here, that involved reactions with microorganisms in surface sediments coupled with some chemical reactions and microbial hydrogenation, and various reactions that these compounds can undergo in the surface sediments. And we were able to pick out and verify the presence of many of these intermediates. This was, to our knowledge, unprecedented, and we were quite excited, wrote it up for a paper which was reviewed, accepted, and came out in Geochem Cosmochem Acta, entitled Sterines and Surface Sediments from the Southwest African Shelf and Slope by Bob Gagosian and John Farrington. Following on in this work in our own laboratories and the labs of other folks in the world, we were able to put together various aspects of the early diagenesis of organic matter. Things that were happening in surface sediments, off in Namibia, in the Black Sea, off the coast of Peru, and other places that we went, that theretofore had not been known and thought could take place only in ancient sediments. Although this might be considered a small advance in our knowledge, it nonetheless helped with understanding how to interpret the records of organic compound analyses in ancient sediments. It was quite exciting. And remember, the whole process is a result of a blooper. Typing in JWF7 rather than JWF6. And then, as is often the case in research, recognizing the facts that nature presents to you when they're staring you right in the face. Thanks for your attention.
That was great. Ross Perot couldn't have done a better job. Are there any questions? Sandy Williams will take any questions. No. Good. Yeah, that was pretty interesting. Okay, the speakers don't actually know what order they're going to go in because I want to keep them all ready at any time. But actually, the next speaker is going to be Dave Aubrey, and you who read Ocean Currents might know him as the Sandman. I hope that's not an indication of how we're going to be doing as far as whether the stimulation level of his presentation. Let's take that as a check. Where is Dave, anyway? He's up there somewhere. Dave and I've known Dave for a long time, ever since I first started as a postdoc here. I've been interacting with Dave, and I've heard a lot of interesting stories about Dave and his research exploits because I used to work closely with Paul Dragos, who went on some of his cruises with him. And one of the more interesting cruises that Paul went on was to Nauset Inlet to deploy Sandy Williams' new sophisticated bass tripod. And they'd never used it in an extremely energetic environment before, and they wanted to study the flow over these sand waves. Well, these sand waves are pretty impressive. They're pretty big. And so they put this bass tripod between a couple sand waves, and then they went down the next day they dove on it. It wasn't that deep, but it was about 4 meters water depth or something like that to retrieve their bass tripod. Well, they had a little surface float, which survived the 24-hour deployment. And they went to the surface float and they swam down the line to recover their bass tripod. And the rope went into the sand and disappeared. There was no tripod there. Well, the sand wave moved over the night, and And all they found was a piece of rope going into the sand. And they spent the next 2 weeks or so trying to, one way or another, to haul this tripod out of the sand. But that's the Sandman's research, and he studies it, but he can't always control it. Anyway, wherever he is, Dave's going to give us an exposition on some topic. I don't know what.
I prefer, I'll just stay, that's all right. Well, good evening, everyone. First, I'd like to start off by thanking the organizers for inviting me here. In fact, I never accepted this engagement. I ignored the engagement, and I never answered them. So they took that as a sign that I had agreed to show up. So they came off and carted me away today. I'd like to introduce, just to embarrass a couple of people in the audience for a change, 3 of our Kazakh visitors. In the back row, we have 3 scientists from Kazakhstan who are studying here for a period of approximately a month, if you can just wave. And as part of a training program for oceanography for some of our Kazakh friends. So we figured this would be a good time for them to learn more about how science is really done rather than what we've been telling them in the laboratory. So tonight I wanted to talk a little bit about polycyclic aromatic hydrocarbons, in particular the distinction between benzoate, pyrene. Oh, those are Farrington's notes, sorry. How do I get those? Okay. I'd like to talk a little bit about a research experience here. As Rocky indicated, I like to do real shallow water oceanography. Some people might not call it oceanography, but fortunately the funding agencies call it oceanography. And I just want to show you what a difficult environment this is and explain to you a little bit about how we came to learn a little bit more about some shallow estuarine systems and how the dynamics of these systems work and some implications for pollutant transport and dispersion and so on in these estuarine systems. So I thought I'd start out with a few slides. We started out this research, remember I'm the Sandman and I like to study beaches. We started out this research by investigating this particular system, which is on the east side of Cape Cod. It's called Nauset Embayment. On the upper left-hand side of the slide is Chatham Inlet, which you've probably heard about and I've talked to, I think, at this very forum in the past. So our particular interest in Nauset Inlet was to study how water and sediment moves out in and out of these shallow lagoonal channels and how it interacts with the marsh surface and with the sand bodies. So one of the first things we did was we went to Farrington. We asked him for the oldest map he had of Nauset Inlet. This happens to be a 1605 map from the early part of his collection, showing Nauset Inlet as it existed when Champlain actually visited the Cape. So if you look, probably on summer vacation, if you look at this slide, you'll see that the slide shows a markedly different character to it than the previous slide. And as a matter of fact, if you compare the shoreline between the uncolored shoreline, which is supposed to be 1605, not 1905, also nimble fingers, it's a requirement for being a scientist, and 1965, you can see that there has been remarkable erosion of the shoreline along the outer Cape. So this is what we went out to study. This is what we were able to convince the funding agents that we wanted to do, was go out in the summertime and do field work out on the beach. Now our field work is rather exotic. We don't have a large stable of instrumentation that comes off the shelf, so we have to invent our own field work and derive our own little instruments. This is a picture of a little very fancy, very, very clever device that measures the beach profile, automatically goes offshore and tells you what the sand level is. It never worked, but it was a very clever little idea. For the studies of Nauset Inlet, we wanted to do some dye dispersion studies. So we asked an engineer here at Woods Hole, Fritz Hess, to recommend some techniques for us. Well, he happened to be a model airplane enthusiast, so he designed a model airplane for us, and we actually put a camera in that model airplane, and we flew all over the Nauset system tracking these little floats that were very cleverly color-coded. We did use color film, by the way. They were cleverly color-coded, so we could look at dispersion processes throughout the inlet. And this is just a map showing that's not blood, that's dye, and along with a few drifters going through the inlet. So this technology wasn't off-the-shelf technology. We had to rely on some of the engineers here at the Oceanographic to try to help us do our experimentation. And we put down a lot of instrumentation to measure a lot of things. Sandy, unfortunately, I couldn't find that photograph of you and I deploying this tripod. Everybody in the back is working, and Sandy and I are arguing about whether the water is going up or down. Fortunately, the technicians just ignore us, and they do their work and let us scientists babble on the side. So we have to put up our own instruments at times. Wayne Spencer has helped us here. He's in the audience, fortunately, with two of his kids, so I can help embarrass him. But a lot of it's just making up new techniques every place we go to try to figure out how to take measurements in water that can be as shallow as dry sand or as deep as 3 or 4 meters deep. And that presents quite a challenge. We use a variety of vessels. This is a vessel called the Zamanique, a little flat-top boat that we modified to deploy our instruments in this shallow water. We did it in good weather and we did it in bad weather, but we always had an excellent crew out there to guide us through thick and thin. It's really tough work out there, I'll tell you. I mean, if anybody lets you think that working on the beach is easy, just look at this picture. That's my hero picture. Okay, sometimes we do biological sampling on occasion, but I'm sure we wrote a paper on this bug, right, Wayne? Okay, it really takes a lot of hard work being a scientist out there on the beach. This is one of our graduate students, Paul Spear. Ben, don't think about doing this next week when you're out in the field. There seems to be a trend here. I'm not quite sure what's going on. This is the pilot, and I hope the plane's safely down, but I don't really see it in the in the background. Anyway, it's really, really hard work, and everybody has to give their all. Well, sort of the blooper that we ran into was we were doing these studies trying to map the evolution of the beach, and every so often we'd look at the tide charts, we'd look at our currents and see how the currents were moving, and we would mispredict exactly when slack currents were or when high or low tide was. This is a picture of our senior pilot, Mr. Spencer, run aground because he miscalculated the tide. That was the first time it happened. He did it again here with even a smaller boat, even less draft. Well, we got a call from Hui Facilities asking for an able pilot to pilot even larger ships. So unfortunately, we gave him Wayne's name, and this was the next situation. Okay, well, accidents happen, and in fact, what really turned out was that in shallow systems like this, we had always assumed that the tide behaves in shallow water exactly as it behaves offshore. In other words, when you have high water offshore, most commonly you'll have slack tide offshore, and when you have low water offshore, you also have slack tide. Well, it turns out that the relationship between high water and low water in these estuarine systems and slack tides really is much different than it is in offshore systems. So this was such an exciting topic for study that for the next 3 or 4 years we spent quite a bit of time and exhausted quite a few graduate students really looking at what we call nonlinear hydrodynamics of shallow estuarine systems, so that the amount of time that the tide floods and the amount of time that the tide ebbs may be different by a couple of hours, and that has very important practical implications for things like sediment transport, for fates of lagoons and estuaries, as well as for fates of contaminants in some of our systems. So the nice result was we've had a couple of good PhDs come out with this nice work, and it really wouldn't have happened had it not been for Captain Spencer. Thank you very much.
Thanks a lot, Dave. It's great having somebody in the flesh, isn't it? That reminds me of a story. Well, I've spent a lot of time out in the field with the guy named Dick Coburn, who a lot of you probably know, who used to be the captain of the Atlantis. And then by the time I got to Hui, he was the captain of the Asterius. And I bought a very expensive piece of equipment to measure currents, and I was studying the currents around Gay Head. And after about 4 months, probably $20,000 worth of Asterius time and about $100,000 worth of equipment, and who knows how much in salaries, I came up with a map of the currents and showed it to Dick Coburn, and he said, "Boy, that looks a lot like what you see in Eldridge." And he was right, and that's how I could tell that I was right on track with. Of course, Eldridge did it about 100 years ago with probably not quite as much expensive equipment, but anyway, yeah, tides are tricky though. They're really tricky. Anyway, the next speaker is Carl Worston. He came to the institution in 1968 as a research assistant with a master's degree in microbiology. He'd been working at Harvard University, but he wanted to come to a place where they do real science. He's presently a research specialist, and he put a few more details here, but I think I'll let him speak for himself, and I think he's going to be talking about bringing your lunch to the deep sea or some such topic like that. Carl.
Well, I also want to thank you for the opportunity to come and share one of our stories with you tonight. In 1968, October of 1968, the submersible that we all know over here as Alvin accidentally sank in about 5,000 feet of water during a launch about 100 miles off of Cape Cod. This happened shortly after I started working here at Woods Hole in the lab of Holger Janisch. Fortunately, no one was hurt during that accident, but the subsequent recovery of the submersible, which was, by the way, an engineering feat in itself, some 10 months later, and the lunch that was in that submarine led to some observations which would have become the basis for the research in our lab for the next 20 or 25 years, and in some aspects it's still going on. I turn on which one is this, left or right, does it matter? Right, there we go. This is the submarine as it sat on the bottom during its sojourn to the deep sea for 10 months, and it was photographed by the Navy ship, the Mizar, before it was recovered. When it was recovered, the lunch that was inside, which had been packed for the occupants that day, was found to consist of bologna sandwiches, apples, and a supposedly unbreakable thermos containing beef bouillon. These materials, when they were retrieved some 10 months later, were found to be in a remarkable state of preservation. The sandwiches, the bread was unspoiled, the inside of the bologna was still pink. The apples, although they had swollen up due to the turgor pressure when they came up, still tasted like a salty apple. We all had a little bit of this in the lab. And the thermos bottle was crushed. It was containing beef bouillon, which was a perfect growth medium for bacteria. It had been inoculated with some seawater, but yet it was unspoiled. By the way, when we saw the state of this guaranteed unbreakable thermos, we wrote a letter to the company, and they quickly sent us a new one, which we still have in use in the laboratory to this day. As I said, all of these materials were in a remarkable state of preservation, which went ahead and spoiled quite rapidly when they were held in the refrigerator in just a few days. So we asked the question, or you might ask the question, how did this observation lead to the science and the research that we were carrying out? Well, the first question we asked was maybe there was some preservative or some sort of chemical that was leaking into the sphere of the submarine that kept this lunch from being spoiled. It was contained in a leather satchel which was open to the seawater, but it was protected from the scavenging animals such as amphipods and the like by not being exposed on the sediment outside of the submarine. it was basically a beautiful experiment in its own. When the Alvin was retrieved, Woods Hole was given the option of either getting the Alvin after it was rebuilt or actually getting the new Navy submarine, the Seacliff, which just had been completed at that time. Woods Hole opted for the former, to have the Alvin after it was rebuilt. We, on the other hand, proposed to the Navy to take the Seacliff, fill it with a lot of new food materials, and sink it as a repeat experiment. As you can imagine, the Navy did not think too highly of this approach. So, in that light, we set out with the help of the buoy group here to do our first experiments with the help of Jimmy Gifford and others, sailing out and putting a lot of packages on some of the deep-sea mooring lines while the Alvin was being rebuilt. After it was rebuilt, we used the Alvin itself to start placing a lot of experiments at what we call permanent bottom stations at 100, 120 miles off the coast here, as well as in the Caribbean, to look at what the effects of high pressure and cold temperatures, which was the probable causative reason why these materials had not spoiled in the deep sea. And we were basically interested in what the activity is of the bacteria that live in the deep sea. Are they specially adapted to those conditions, and what is their rates of growth and activity? At the same time that we were conducting our experiments, Fred Grassley, who is now at Rutgers but was here at the time, carried out very similar experiments at these stations along with us, looking at invertebrate animals. In summary, the observation of the preservation of this lunch led to the conclusion that much of the activities, particularly the microbial activity, as well as some of the higher animals, in the deep sea is greatly repressed by a factor of ranging from 10 to 1,000-fold compared to what would be occurring in your coastal shallow waters and the like. It also happened to be at the same time that a lot of questions were being posed about using the deep sea as a repository for organic waste and deep sea dump site. And in fact, what this would have done would have put a lot of material perhaps out of sight and out of mind, but it was basically would have been collected in the deep sea and not been recycled in the traditional sense of turning over in the soil or in the shallow water environment. Then over the next several years, we proceeded to employ the help of the engineering staff here at the Oceanographic as well as the machine shop to build a whole suite of pressure retaining equipment which allowed us to go to sea and obtain samples from the deep sea to ask more sophisticated questions. This has just recently culminated in the construction of what we refer to as a high-pressure chemostat, which allows us now in the laboratory to mimic the conditions of the deep sea as closely as possible by maintaining high hydrostatic pressure, very cold temperatures, and a constant but very limited supply of food source for the bacteria that we have isolated from the deep sea. Basically, I consider myself very lucky to have been involved in these very early observations, and then even more so to subsequently use the tool, the Alvin, for many years, which was basically responsible for bringing these questions to light in the first place. I'm going to close with a very brief recollection of what I think occurred on my first dive in Alvin after it's been refitted in 1970. This was out at one of the permanent bottom stations that I referred to that we had set up off the coast here, in which we revisited many times over the course of years. On this first dive, I think the pilot was actually Ed Bland, who was the person responsible for avoiding a disaster on the day that the submersible actually sank. Well, we were proceeding along the bottom at a normal rate, looking for a nice place to set some of our experiments on the bottom, when Ed literally put on the brakes on the submarine. And putting on the brakes on the submarine means reversing propulsion, and in so doing, on a silty and sediment bottom, he just stirred up this whole cloud of sediment, couldn't see a thing. And while he was doing this, he exclaimed at the same time, "Uh-oh, it's a hedgehog. It might be alive." Well, now, being a biologist and him saying hedgehog, I'm thinking animal. Big animal, small submarine, uh-oh. Well, I sat there and controlled my fears for a few minutes, and as the sediment settled down, he let me look out of the front window. And in fact, what the hedgehog was basically a bomb of sorts sitting in the bottom. And these were bombs that were used, or explosive devices that were apparently shot off of destroyers to try to locate submarines during World War II in clusters. And if they did hit anything, they would go over and drop depth charges. and many of them, as you can imagine, did not hit anything but just sank and impacted on the bottom without exploding. We mapped quite a few of these in the area of our bottom station, and we're very careful to avoid them over the course of our dives. But it nevertheless brought home a message to me at the time. This was my first impression of the deep sea, and also the first impression of man's impact on the deep sea, on how long-lasting and significant it can be, and it drove home to my mind how careful we have to be in any of our decisions in terms of this environment, since it is a fairly fragile environment in the sense that once we have perturbed it, it's very difficult to make changes in those situations. So, thank you.
Thank you very much, Carl. I wonder what would happen if it had been a roast beef sandwich instead of a bologna sandwich. The bacteria might just have good taste, you know. We're going to have a little interruption in our formal schedule, and Dave Mash is going to add to Carl's anecdote with a related event that happened 4 years later or so.
This story I'm going to tell isn't directly science, but it involves science and it involves experiences that everybody's had that's gone to sea very often and used heavy equipment on a boat. In, I believe it was 1972, when the GSX expedition, we were on the Nore, somewhere below, it was near Antarctica anyway, and we were doing sampling and bottom photography using a giant Edgerton camera and a new set of equipment for us, which was this great, Ralph Vaccaro could tell me what it was called, I think. It had a great ring, a rosette of water sampling bottles. Cost a great deal of money and was sending back electronic information that we used to all measure manually. This thing cost a great deal of money, and it would be lowered down to the bottom of the sea where photography would go on and sampling would go on. It was the middle of the night, maybe 40 degrees outside. The boat was slatting around, and it takes a long time to lower an instrument to the bottom. So a man sits at the winch, and the man sitting controlling the winch this day was a guy that many of you may remember, Fat Eddie Broderick. He used to be the cook on the Lulu, and he got so big he could hardly get in and out of the Lulu. And his doctor said, either you give up cooking or you're going to die. So he became an ordinary seaman. And finally an able-bodied seaman, though a large-bodied seaman. He was at the winch controls, and this is across the deck of the Nore, the fantail, and the wire that holds the very heavy equipment goes through a big ship hung out on an A-frame. And the boat's going up and down, and every once in a while a kind of alarming slam will happen when you hit up against the tension on the wire. There's a great deal of strain on this wire. And he'd been sitting there for a couple of hours. The thing came up once and slammed, and the wire broke. So when this happens, panic goes through the ship. Everybody was asleep. The chief scientist was a man whose name I don't recall. It's probably good. Because he wasn't from the Woods Hole Oceanographic Institution. I'll go that far. He wasn't very well liked, and he was very excitable. He showed up, and everybody else showed up, and as you all know, the first thing you do when you lose anything in the deep ocean is you run to the side of the boat and you look at the water. It happens without exception. 3 or 4 more people come on and say, oh my God, we've lost the gear. They all walk to the side of the boat. And everyone does it. No matter how many times you've been out there, and at night all you see is this cone of light that disappears. Everybody looks and goes. Meanwhile, this wire, when it broke, had snapped back across the deck, and Eddie was sitting on a wooden bench. He's about this wide. The bench was about this wide, and beside him, the back of the bench was wood. A chunk of wood about 4 inches deep, an inch thick, had been torn out by the cable as it snapped back and then recoiled. If it had hit Eddie, we probably would have had 2 large pieces. But Eddie seemed strangely calm about the whole thing. The chief scientist showed up, and he was mad at everybody. He just was wild with anger or fear, disappointment. He said, "Do you realize what that cost?" The camera alone is $55,000, and the rosette's 80. That's why aren't you upset? This guy was nearly killed. Still didn't seem upset. Eddie said, well, he said, do you remember when the Alvin was lost? He said, I was one of the guys holding one of the lines. We launched the Alvin. He said, hell, that was almost $3 million. That's the extent of the story.
Thanks a lot, Dave. Our next speaker is the hostess of this event, Gigi McDowell. She's a zoologist interested in how animals adapt to changing environments, including contaminated habitats. And her accidental discovery relates how contaminants alter the reproductive biology of mollusks.
Well, these guys are a tough act to follow. I'm very serious. I'm not very comedic, so I don't have many jokes. But I do attribute my blunder to John Farrington, who is not here to defend himself. So it was a great discovery that I made based on a blunder of John Farrington's experiment. And if I could have the first slide, please, Tim. A number of people have been studying the New Bedford Harbor Superfund site for over 15 years now in trying to understand the fate of contaminants from the Superfund site and its effect on marine organisms within the harbor. And in the early 80s, John Farrington, Bruce Tripp, Hovey Clifford were doing some mussel watch studies in the harbor. And they had a series of mussel cages deployed throughout the inner and outer harbor of New Bedford, looking at the accumulation of PCBs and other contaminants within Middleus edulis, the blue mussel. A couple of years later, John asked if I was interested in looking at some of the biological processes going on within the organisms, and so we combined studies of interest of accumulation of contaminants and the biological effects of those contaminants on these mollusks. To convince you that indeed they are highly contaminated, there's signs posting within those habitats of no fishing, no swimming, no shellfishing, no lobstering, because of the high levels of PCBs within these tissues. Now, my part of the investigations that I initiated was actually to look at how contaminants accumulated in the mussels, particularly in their fat stores, their lipid stores, how those lipid stores were used in the reproductive cycle, and what was the consequence on reproductive output. And this was about 1984 and 1985 is when I conducted my field experiment. And prior to this time, there had been a number of laboratory studies on the effects of contaminants, polyaromatic hydrocarbons, petroleum hydrocarbons, other types of lipophilic contaminants, including PCBs, on reproductive cycles of mollusks. And most of the laboratory results suggested that there was a disruption in the ability of the organisms to feed, which resulted in less energy accumulated and thus less energy available for reproduction. And that seemed to be the way we were clicking along with our experiments, and we found some disruptions in feeding, disruptions in respiration, disruptions in energetics, and we came up, that there did seem to be some perturbation in the reproductive cycle. And then John and Hovey came back from a cruise one day, and they discovered a cage that they didn't tell me about this one until later on, that they had lost in 1983. A cage that had been deployed in New Bedford Harbor, and the only remnants of the cage was the buoy attached to it, which had some shotgun pellets in it. And some of the dangers of working in New Bedford Harbor, in addition to the contaminants, are also sort of the wild recreation boaters and other activities on the weekend. And someone had taken a shotgun blast to the buoy and sunk the cage. And Hovey and others and Bruce Lancaster and Dale Levitt were diving in the harbor to retrieve some other samples, and they came upon this cage. So they brought the cage back to the lab, and John was very excited about this. You know, here, not only did he have natural populations that had been exposed, but here he had this cage of mussels that had now been down in the harbor for at least 2.5 to 3 years. A great number of animals had survived and wanted to look at the chemical contaminant burden in these animals, and he decided that he wanted to look at males and females to see if there was a gender distribution and gender differences. So since I was the only one in the group who knew anything about the reproduction of animals, they were looking, they'd open up all the mussels, and they would just look at color and say, well, this must be a male, this must be a female. And I said, that's not the way to do it. So I started making all these slides to determine the males and the females, put them in different groups, and go on through the extraction procedure. And everybody's getting real excited about all this. Hurry up, you know. And I find the males, and I had a nice big pile, you know, of male mussels ready to be processed. And I couldn't find any females. And I go, do more, hurry up. And I said, you know, now you're trying to do all this careful microscope work with 4 people breathing down your neck. So the conclusion of the study was that there were no active females. There was not, within 100 animals, there was not one sexually mature female. All the females had gone into an indiscriminate stage of determination. There was no active gametogenesis. There was no active ovarian development in any of the mussel populations. And the sex ratio of the original sample that was deployed in the harbor was about 1:1. So there were both males and females deployed originally in the cage, and yet there was no ovarian maturation of any kind. In fact, that there was no reproductive activity of any kind within these mussel populations. And that has set us off on, probably the last decade of research to look at the mechanisms by which contaminants interfere with ovarian maturation and the reproductive, particularly in the females of molluscan populations. And in some ways, people say, oh, what's an early discovery of estrogen disruption in the environment? And in some ways, molluscan systems are quite different than mammalian and other vertebrate systems. They're not known to actually have any kind of estrogen function in terms of the maturation of the gonads. But indeed, they do have some hormonal regulation of yolk production and yolk synthesis. And this was one of the first field observations that there was sexual differentiation between males and females in this phenomena, and has really led to some exciting discoveries for us on the basic control of reproduction within the female bivalves, as well as some of the perturbations within those systems. So that came from a blunder or an accident of someone losing the cage or deliberately destroying the cage in the environment, but the discovery of that cage later on, and the enthusiasm to start looking at the samples and to start looking at the differentiation of chemical accumulation in the samples, but led to the discovery that there really was a differentiation in biological response as well. So.
Thank you, Judy. Can you imagine 100 males and no females? I went to Dartmouth College, so I actually can imagine that. Oh, by the way, how many people, let's have a show of hands, how many people noticed that the first slide was in backwards? I was thinking that was maybe the Southern Hemisphere version of, actually I was thinking maybe it was on purpose, but anyway, the next presenter is going to be Stan Hart, who didn't want me to say anything about him because he's hoping that he could get up and get back into his seat without anyone really noticing who he was. He's actually a geologist, a senior scientist at Hui, and that's all I'll say. Use that. Okay, I have one prop.
What's on the back of your shirt, Sam? I'm not going to turn around because I'm trying to be incognito. This is my own personal example of serendipity, which in everyday speak I think means dumb luck. There's a label on it. It says the S.R. Hart Award, Scientific Advancement Through Misidentification. Some of my graduate students at MIT gave that to me quite a few years ago in honor of a blunder that I made as a graduate student. To understand the setting for that, we have to go back to the mid-50s when I was a graduate student. That's not as far back as 1605 when Champlain and John Farrington were graduate students. In those days, geologists were particularly interested in determining the ages of rocks. That was the big sort of new thing. And one of the principal ways that they did that was to take a mineral, in this case biotite, it's a mica, which has a lot of potassium in it, and one of the isotopes of potassium decays to argon, so if you take the biotite and do some fancy chemistry, measure the potassium in the argon, you can tell how old the biotite is. And that was a very common way to determine geologic age in the mid-50s. Now, I set out on a thesis project to try to figure out why biotite didn't always give the right answer. We knew that there were times when it worked pretty well and times when it didn't work very well at all. So I decided on a thesis with my advisor, Pat Hurley, at MIT, which involved going out to Colorado and finding an area of very old rocks that had been intruded by a very young sort of volcanic igneous melted rock, which heated the surrounding rocks. And the hypothesis was that this heat would heat the biotite in the surrounding rocks enough to let the argon diffuse away and sort of screw up the clock, as it were. And so I went out to Colorado, found this young igneous intrusive set in the midst of very old Precambrian country rocks, and the idea was to start at the edge of the intrusive and collect samples at different distances until one got so far away that there could not be any possible thermal effect. Well, I spent 3 weeks there, collected a car full of rocks, literally, all meant to have this black mineral called biotite in them. Well, I got back to MIT and started grinding up the rocks and to separate this biotite for the analysis, and rapidly realized that what I brought back wasn't biotite. It was black, but it wasn't biotite. And of course, I had done an undergraduate degree in geology at MIT and a master's degree at Caltech in geology. And the one thing they teach geologists big time is how to identify minerals. And the thought that I could bring back literally 2,000 pounds of rock, which was supposed to be biotite, and have zero biotite was fairly discouraging. And I literally gave up the idea of doing that thesis, and I went and sort of sat in the library and stewed in my juices for a couple of weeks trying to think of something new to do that didn't involve any field work. But then I recovered some sort of sense of intuition or something, and I decided to start thinking about, by this time I had figured out the black mineral I brought back was hornblende, not biotite, so I had 2,000 pounds of hornblende. Ostensibly, hornblende doesn't have any potassium in it, so it's no good for what I intended to do. But I did some library work and in fact found some sort of mediocre chemical analyses for hornblende that suggested that it might have a little potassium in it. I thought, okay, well, in desperation, I went and measured some, and while it didn't have as much potassium by a factor of 10 as biotite, it in fact had a little bit. I thought, well, I'll try dating this stuff just because I have a lot of it. And it turned out I had to tune up the vacuum system in the mass spectrometer to deal with the smaller amounts of argon that were present in a mineral that had 10 times less potassium than the biotite. But in the end, it worked unbelievably well. And I mean, I eventually, a few years later, went back and did manage to collect some biotites from the same area to bring back and compare. And it turns out that the effect of the heating from this intrusive was extremely serious for the biotite, out for several miles. Whereas the hornblende, once you got out even 10 feet from this contact, was literally pristine. It carried its original Precambrian age. It hadn't been reset. It hadn't been disturbed in any way. And basically, I had stumbled onto a new geochronometer, which has been for many, many decades a geochronometer of choice to this day almost. And it's all because when I was an undergraduate at MIT, they didn't teach me how to tell biotite from hornblende. Thank you.
Stan, you've got a clip. You still have a... They're trying to steal it. They don't pay you enough, huh? Stan broke the microphone. I'll have to hold it. I was a geology undergraduate major. A friend who wasn't a geology major, she obviously wasn't because she wasn't sympathetic, handed to me and a friend a piece of tar, well, asphalt, and asked us to identify it. And we got our acid out and our magnifying glass. We had 3 or 4 different theories. She got a big kick out of us not being able to identify a hunk of asphalt. Okay, I think, is Eben Franks here? Okay, in that case, our last speaker of the evening is Sandy Williams, who has regaled the Applied Ocean Physics and Engineering Department for years with his insatiable curiosity and inventiveness in all sorts of scientific endeavors. He once put his head inside a, what do you call those? Magnetic field, Gauss. Anyway, an extremely powerful magnetic field to figure out what it might do to his head. He never realized the effect that it had, but the rest of us have. And he'll go on to demonstrate the incredible creative powers that come from putting your head in two
Yes, the
intensive
magnetometer magnet had a very strong effect on pliers and gas discharge tubes, and I thought the Hall effect at least might distort thinking, but if it did, I never noticed it. Well, I actually relish my disaster stories, and I have a reputation for telling disaster stories, but when I went over the bloopers that I have experienced frequently and sorted them by those things that did have a serendipitous outcome, I realized that was a much smaller set. You often learn things like, well, you should really mouse the latching hook before you put an instrument over the side if you intend to retrieve it again, or it'll just lift off the hook and fall into the sea. And there makes it a very good story to see your instrument hanging by its feet upside down 30 feet below the ship because it just lifted off the hook. But that didn't really lead to serendipitous discoveries. I think that there's a class of blooper that's sometimes serendipitous that derives from the attempt to make something work where if you knew better, you would never have attempted it. And I think Columbus's discovery of America falls into this class. Educated people of his time knew the diameter of the Earth, and they knew how far away India was, knew that you'd never get there going the other way around in a sailing vessel. But either through ignorance or obstinacy, Columbus did go the other way and thus discovered an unpredicted continent. I owe several minor successes to just such ignorance on my part, and in retrospect, my attempt to do what a better informed engineer or scientist would advise was impossible yielded unexpected benefits. The platitude might be, don't be too sure you know everything, try it, you might learn something. I did not discover hyperbaric microbiology from a serendipitous accident, but I did discover salt fingers in the ocean, which is an important but obscure process that mixes heat and salt downward across the thermocline, and I discovered that through ignorance of the optical index of refraction of those cells. In the laboratory, counterflowing cells of salty and sugary water can be made by floating a dilute sugar solution over a concentrated salt solution. And then if you shine light horizontally through the tank and project that light on a screen inches or feet away from the tank, you see at the interface fine vertical shadows. These are the shadow graphs of sugar fingers. Now in the ocean, salt acts as the sugar in the laboratory experiment, and heat, or really cold, acts as the salt does in the laboratory to stabilize the flow. So in the ocean you have warm salty water over cool fresh water in most of the ocean because of evaporation and heating at the surface. Because it could be seen in shadow graph in the laboratory, I thought, well, let's look for salt fingers in the ocean using an optical technique. But the index of refraction anomalies between the up and down going fingers being much less than in the laboratory required a much more sensitive detecting system, and I made one out of a combination of a telescope and a microscope that had such sensitivity. And when I deployed this system in the Mediterranean outflow, the region in the Atlantic outside the Mediterranean where the warm salty water from the Mediterranean intrudes into the cool, relatively fresh Atlantic water, I found vertical bands just like in the laboratory. Well, delighted that the technique worked, that there were salt fingers right where they were expected, and that this obscure and so far hoped-for phenomena actually existed, I turned to writing the paper describing the instrument, technique, and observations. And Izzy, you were there. It was miserable, but I did it. I had lost the instrument by that time. Izzy and I were on a cruise together, and she said it damn near ruined our marriage. But trapped on a ship with a computer and nothing to do, I wrote the paper. And in fleshing out the part of the paper where I spoke of the index of refraction, I discovered that the effect of heat and salt nearly canceled, and that salt fingers, in fact, should be invisible. But not having done the careful analysis in advance, I hadn't given up. I hadn't tried to find salt fingers acoustically or by conductivity signal as others had proposed and failed to do. In fact, my ignorance saved me from giving up in the face of hopeless odds, and I prevailed. In retrospect, I believe that the internal structure of salt fingers may have saved me. The distribution of temperature across a finger may be different from that of salt. The temperature may vary more uniformly. The salt diffusing more slowly than temperature may be more square. And so there's sort of a second order effect that may have made salt fingers visible even though the average effect of the temperature and the salinity would cancel in the index of refraction. Well, I advise my students and other colleagues to try it when it's not clear if whatever they think of will really work. The reason is that things are often not quite as we expect them to be. And this is where a certain fraction of bloopers or blunders serendipitously produce results. When one thinks of a new idea, it often helps to do a calculation, to go back to a textbook to see if you've got the physics approximately right. But after that, one should build it and try it before going to the library and finding out what everybody else has tried and giving up because it won't work. And that really is the major point I wanted to make, although there are a whole list of blunders I have made, even a few of which have led to serendipitous results. Thank you.
Okay, Sandy was the last of our scheduled speakers, and I guess I'll now invite anyone from the audience who is dying to share some experiences of your own that have or haven't led to incredible scientific breakthroughs. We'd be happy to take on any comers. I didn't think there would be that many brave people. Okay, well, in that case, I think, well, Judy wanted me to point out that those of you who didn't get copies of the article.
The article on Teflon that was in Discover magazine. Teflon was an accidental discovery. It's in the October '96 issue of Discover. Or if you ran out of copies, but you see Sherry or Tracy and give me your name, you'll send me copies.
And I guess I'll just close by saying that if we knew what we were doing, it wouldn't be research.